swim away from it or move away from it. Food is still probably pretty abundant on the edge, so they may still be growing logarithmically on the edge. But those on the center are probably in a different place in the growth curve. Now, when you look at growth in a broth culture, you don't really have that same dynamic. The distribution of cells is pretty evenly, the distribution of waste is pretty evenly, the distribution of nutrients pretty evenly. So the depletion of all those things and the accumulation of waste all going to happen pretty evenly throughout the tube. Now, of course, settling occurs, and so probably a little more dire at the bottom of the tube than near the top. But for the most part, the microbes in the test tube are going to be all in the same place in the growth curve, as opposed to where the microbes in the colony might be. Yes?
It might vary from species to species, but do they tend to divide when conditions are optimum or when the conditions seem to change, where there are not as much nutrients or...
They are always going to be dividing when conditions are optimum. They're always going to be dividing. That's what they want to do. That's all bacteria cells want to do, is just divide. And so their goal is to divide, if you could say they have a goal. Meaning then that they're going to use the nutrients that are there. Now, if there are not enough nutrients there, they're not going to divide. And that's what will happen. Either they'll have enough to produce replicate the DNA and the structures and stuff to divide, or they won't. Okay. Now, sometimes, depending on the microbe, and we'll see this on Monday, if the microbe is a spore former, it can interpret the environment as being not a good place to be, and instead of trying to divide, form spores instead. So you get endospore formation instead of division. And so if we were to, if we assume then this colony is a spore former, an endospore former, if we were to look at these cells here, they probably have already started to form spores, whereas these cells over here maybe not so. Okay, we talked about environmental factors, temperature, pH, water availability, oxygen requirements. Be familiar with all of those factors. Understand or know the terms. I can easily, give you a definition and ask you to provide the term. So I might say prefers a temperature between 20 and 40 degrees. Mesophile. I might say prefers an acidic condition of less than 5 pH. Acidophile. Okay, so these could be matching questions that I might give you. Grows in the presence of oxygen but doesn't use it as a terminal electron acceptor. That's tricky.
Is it an obligate anaerobe or is it a facultative anaerobe?
It would be an obligate anaerobe. No, because an obligate anaerobe implies that it has to be in an anaerobic condition. See, this said it grows in the presence of oxygen but doesn't use it as a terminal electron acceptor. Aerotolerant anaerobes, also known as the obligate fermenters. I forget already. No, really, I have. It grows in the presence of oxygen, but doesn't use it as a terminal electron acceptor. Aerotolerant, or obligate fermenter. And the group of microbes that fits that were the lactic acid bacteria. Way back in chapter 11, second day of class, we talked about those guys. Nutritional factors, we talked a little bit about those. Understand the term fastidious. It means it's a microbe that needs several growth factors. Enrichment culture. Know what an enrichment culture is and why we would use an enrichment culture. Chapter 5 looked at the control of microbial growth. And so again, terms that you need to learn: sterilization, disinfection, antiseptic, pasteurization, things that you've probably encountered before. Again, they lend themselves well to matching. So I can give you a definition, you match the term with it. Know the different types of control that we talked about. We talked about physical control and chemical control. Physical control, we used heat, two types of heat, moist heat and dry heat. Understand why or when you would use one versus the other. When would I use a moist heat versus a dry heat? If you're autoclaving media, you will want to use moist heat. Or if you want to sterilize media, you want to use moist heat because you don't want to evaporate the water out of the media. If you put media into an oven, you can sterilize it if the oven's hot enough and you leave it there long enough, but you'll probably evaporate out all the water, and so that doesn't work. You would use an oven though if you had delicate instrumentation that you couldn't get wet, glassware that you don't want to get, put in the autoclave. Those things can go in the oven and be fine. We talked briefly about different types of radiation, mentioned gamma, UV, and microwave. So with regards to chemicals, your book has a list of chemicals, and I didn't talk about all the chemicals in class that your book talks about. I only brought in a few household cleaners. Those are the only ones that I'm going to ask you about. Okay, so know how those cleaners work or operate. We talked about alcohol, hydrogen peroxide, I didn't bring in sodium hypochlorite or bleach, but we did talk about halogens or bleach. We talked about quats, the quaternary ammonium chloride molecules, or cleaners I should say. Those work to loosen the surface tension on between the microbe and wherever they might be so that they get wiped away or cleaned away, in addition to disrupting the cell membrane. Okay, so that brings us to chapter 6, metabolism. Okay, so you need to know some detail. You don't need to know all of the enzymes and all of the different steps that take place in the pathways, but you do need to know the gist of the pathways. For instance, glycolysis. We take a 6-carbon molecule, glucose, and we break it down into 2 3-carbon molecules, pyruvate. And in doing so, we generate 2 ATP and some reducing power. That's it, really. That's what I want you to know about glycolysis. You also want to know where it happens, though. It happens in the cytoplasm of the bacteria cell, and it also happens in the cytoplasm of the eukaryotes.
What is a pyruvate?
I'm sorry, pyruvate.
What is that?
That's a 3-carbon molecule at the end of glycolysis.
So it's a molecule in glycolysis?
At the end of glycolysis.
So it is 2 ATP and what reducing?
Reducing power. The NADH, that molecule that is produced is what we've described those as having reducing power. Okay, meaning it's got a high energy electron that it's going to take to the electron transport chain and hand off.
You want us to know the net ATP and
The what?
net ATP?
No, I don't want you to know the net ATP or the net NADH that are produced necessarily. I won't ask you those questions. Just the gist of the pathways, what's going on in each of those pathways. The tricarboxylic acid cycle, okay? There's a transition step where the pyruvate has to be broken down into a 2-carbon molecule before it can enter into that TCA cycle. And then the TCA cycle has to turn 2 times to complete the breakdown of a glucose molecule. And what do we get from the turning of the TCA cycle? What do we get from that? Okay, we don't need to know the number necessarily, we just need to know we get some ATP, and we get a lot of reducing power, don't we? We get 3 NADH molecules, and we get an FADH molecule for every pyruvate that turns. So we get a lot of reducing power from that.
Does that include some energy or not?
Well, the reduced power has high energy electron, yeah. So now, we've pretty much done with the breakdown of the glucose molecule. The next step is to use all of that reducing power that we have trapped in those NADH molecules, and that's where the electron transport chain comes into play. Now back to our TCA cycle. In bacterial cells, it's happening in the cytoplasm, but in eukaryotic cells, pyruvate molecule has now moved from the cytoplasm into the mitochondria. And so the TCA cycle takes place in the inner lumen of the mitochondria. So now we've got the reducing power. This is where the cellular respiration happens. The molecule transfers the electrons to those protein complexes embedded in the membrane, and we pump hydrogen ions out of the cell, establishing our proton gradient. Okay, and that could be an essay question as well. Describe the proton motive force. You know, what is the proton motive force? Why do we establish it? Okay, it's a gradient of hydrogen ions outside the cell, high concentration of ions outside the cell, and because of that, they're going to want to naturally diffuse into the cell, but they can't get across the membrane, so we have these little gates that we use, and as they diffuse across those gates or through those channels, the energy that we get from them moving in that direction is captured in ATP. You should know what happens if there is no terminal electron acceptor. What happens when that 4th protein complex cannot pass on? Sorry? Fermentation, yes, because the whole system gets shut down. The electron transport chain gets shut down if there's nothing there to receive the electrons. The TCA gets shut down, and glycolysis would shut down. But if that happens, the cell's going to die because there's no energy being produced. And we can't let that happen. So what happens then is pyruvate becomes that terminal electron acceptor. Pyruvate accepts electrons from NADH. Thus, NADH becomes oxidized and now available to receive electrons, and that allows then for glycolysis to continue.
Is there a substitute TEA in the electron transport chain?
A substitute what?
Terminal electron acceptor. So like, you know how you get the oxygen and half the oxygen that goes out? Isn't the oxygen?
In aerobic organisms, the oxygen is a terminal.
If there's no oxygen to be the terminal electron acceptor, there's no pyruvate in that part of the reaction. It's already been mixed up over here, producing either fermenting or doing the glycolysis, right? So what's the substitution for oxygen then?
No, there's still pyruvate available.
There's still pyruvate?
Yeah. Because you got to realize glycolysis is continuing. Even though you've broken down some of the, say you've got 100 molecules of glucose, right? And you're breaking those down, and maybe 50 of them have gone through the TCA cycle, okay? And then you run out of oxygen, okay? That glucose is going to continue to break down if you can take the pyruvate, some of that pyruvate, and recycle those NADH
Right,
molecules.
via fermentation.
Right, via fermentation.
Okay, so what happens once you get, I understand that part of it, but does that continue within the electron transport chain?
No.
Okay.
No,
so what
pyruvate
happens-
does not serve as a terminal electron acceptor at that point.
Right, so
Okay.
that's, okay, so what's the substitution in the electron chain? Hydrogen?
There is no substitution. If oxygen's not there, the electron
It's not
transport
enough.
chain
Thank you.
does not go.
Okay.
Unless you're an anaerobic microbe that uses nitrate or sulfate as your terminal electron acceptor. Then respiration will continue in the absence of oxygen. But then you wouldn't use oxygen as a terminal electron acceptor.
Anyhow, exactly.
You said that respiration occurs in the movement of the mitochondria, correct?
Respiration occurs on the inner membrane of the mitochondria.
Okay, so when it's shuttling protons out to create the gradient, do those protons just automatically go straight all the way out of the cell?
No, good question. So here's our mitochondria, and here are where our proteins are embedded, and here's where the TCA cycle is taking place. Those hydrogen ions are actually accumulating right here in this matrix between the two membranes. So no, they don't go all the way out of the cell.
Okay, so how do we establish the gradient then?
Well, across the membrane, because now you've only got a few in this inner part, and
Oh,
you've
just
got
in
many.
the mitochondria you're talking.
Yeah, just in the
Okay.
mitochondria. So this is the big cell.
Yeah, yeah.
Okay.
Okay, so you only need the proton gradient